Now, we have already told that regulation of enzyme activity can be done via 5 means. That is your allosteric modification, then your covalent modification, then is your regulation of enzyme activity at genetic level, then is your proteolytic trimming, lastly the compartmentalization. So these are the 5 means by which we can regulate the enzyme activity. Now here, the regulation of enzyme activity at the genetic level. It can be via 2 measures. One is induction, another is repression. So what is induction? There is increased synthesis of enzyme and some inducer molecules are there which relieve the repression on the operator side. So there is increased synthesis of enzyme. Okay, so some repressor molecules are there. They inhibit the enzyme synthesis and some inducer molecules are there. How they act? They relieve the repression and they increases the enzyme synthesis. So these two, that is our induction and repression, these are the two main things which operate the regulation at the genetic level. Now, the induction. This is affected through the effect of derepression. Now, the inducer molecule will remove the block on the biosynthesis of the enzyme molecule. So there are in the synthesis, during the biosynthesis, the inducer molecules, they remove the block. So there is increased amount of enzyme synthesis. So there is increased concentration of the enzyme. So the enzyme activity increases. And the classical example is the induction of synthesis of lactose utilizing enzyme in bacteria when the media contains lactose. This is our lac operant concept. And another example is our glucokinase, which is induced by insulin. We all know that glucokinase is the first enzyme of glycolysis, and this enzyme is under the regulation of the hormone insulin. Insulin acts as an inducer of the enzyme glucokinase. Now see, this is a DNA for the synthesis of the lactose utilizing enzyme. So what are the lactose utilizing enzymes? That is galactosidase, it is beta galactosidase, galactoside permease and galactoside transacetidase. In this picture you can see the 3 genes, red, orange and violet. So the red, lac Z, this portion encode the enzyme beta galactosidase. Lac Y, this portion encodes the galactoside permease, and lac A, this portion encodes galactoside transacetidase. And here students, you can see the other portion, that is our operator site and the promoter site. In the promoter site, usually the RNA polymerase binds, and lac I, it is the regulatory portion. So if I consider the DNA, then there is a regulatory portion, a promoter portion, and operator portion. And downstream to it, the structural genes are present. Here, lac I, this is the regulatory portion, then the promoter portion. Here the polymerase binds, the operator site which regulates where the inducers of the repressor molecule binds, and the structural gene, that is the lac Z, Y and A. So this is the basic picture of our lac operant. Now what happens when the inducer molecules are present? See in the next slide. Now see, in this slide you can see the lac Z gene, lac Y gene, lac I gene, lac A gene, lac Z, Y and A. These 3 genes you can see. The operator site you can see, the promoter site you can see, and the lac I gene, that is the regulatory portion, you can see. Now when the media content no inducer, means no lactose is present, then there is no need of synthesizing this lactose utilizing enzyme. Then what happens? This lac I gene, their transcription happens and some repressor molecules are synthesized as a monomeric form. Now this repressor molecule now form a tetrameric form, and this repressor molecule now can bind at the operator side. This tetrameric repressor molecule can bind at the operator side. So now they block the operator site. Now when the RNA polymerase, now as they block the operator site, the RNA polymerase cannot code or cannot transcribe the downstream genes. So there is no synthesis of the LAC-Z, Y and E, that is the beta galactosidase, galactoside permease and galactoside transacetylase enzyme. So there is no metabolism of lactose. So there is no lactose. So there is no need of synthesizing this enzyme. Okay, when there is no inducer. But what happens when the inducer molecules are present? In the next slide, we will discuss it. Now see, in this slide, when the media contains lactose, then now to metabolize this lactose, the enzyme must be there. So what the lactose does, the same thing happens, the LAC-I portion, they synthesize the repressor molecules. But here when the media contains lactose, the lactose molecule act as an inducer and they bind to the repressor molecule. Now the repressor cannot form the active tetramer. So the repressor molecule cannot bind at the operator site. So the operator site becomes free. Now the RNA polymerase can bind at the promoter site and they can transcribe the downstream genes and there is increased synthesis of the enzyme beta galactosidase, galactoside permease and transacetylase. So this is a typical example of induction. See, in the previous case, in the previous slide, we have already discussed when there is low lactose in the media. No lactose means no need of synthesizing this enzyme. So there is the repressor molecules are synthesized, they form the tetramer, they bind at the operator site and they prevent the RNA polymerase to the downstream transcription of the other genes. But when here the operator site is occupied by the repressor molecule. But when the media contains lactose, then this lactose molecule, they inhibit the repressor subunit. So the repressor cannot form the tetrameric subunit. So the operator site, they cannot block the operator site. So the operator site is free now. Now the promoter site can be occupied by the RNA polymerase and the RNA polymerase can easily transcribe the genes for RNA synthesis. Clear? This is your repressor. So what the repressor does, they decreases the enzyme synthesis and also they decreases the enzyme activity. So there is a basic difference between the repressor molecule and inhibitor because inhibitor also decreases the rate of kinetics and repressor also decreases the rate of kinetics. But there is a basic difference. What is the difference? The inhibitor, they act directly on the enzyme. They may bind they may be as a structural analog of the substrate and bind at the active site. They may bind at another site of the enzyme or they may bind at the enzyme substrate complex. But the repressor molecule, they does not act directly over the enzyme. They act at the genetic level. This is the first difference. Next is the inhibitor molecule, they after binding with the enzyme, they causes some conformational changes within the enzyme. So the binding affinity of the enzyme towards the substrate is decreased or lost. But the repressor molecule, what they does, they decreases the enzyme synthesis. So inhibitor does not decrease the enzyme concentration, but repressor molecule, they decrease the enzyme concentration. Okay. And thirdly, the inhibitor, their action is very rapid because they act directly over the enzyme. But the repressor, they act at the genetic level. So a lag period is there. Okay, so these three are the major differences between the inhibitors and the repressor. Now, this repressor molecule, they decreases the enzyme synthesis. Now, one classical example is inhibition of ALA synthase, that is amino levulinic acid synthase by heme itself. Okay, in the previous lectures, I have told you about heme synthesis. Here, the first step is the succinyl-CoA and glycine, they combines and the enzyme comes ALA synthase. So ALA synthase is that is the key regulatory enzyme of the heme synthesis. And this enzyme is in direct regulation of heme via this repression. Now see students, this slide. In this slide, you can see the regulator, the promoter, the operator, and the structural. These are the four portions of a DNA. And here the structural, this is the gene for allosynthetase. So the structural gene is allosynthetase. Structural gene is for allosynthetase. Now, what happened? The regulatory portion, they synthesize it, they transcribed on mRNA. From this mRNA, we get some apodipressin molecule. Now, these apodipressin molecules are unable to bind with the operator site. But here, the heme molecule act as a co-repressor. They bind to the aporepressor and they form the holorepressor. So aporepressor plus the co-repressor, this is the heme, they form the holorepressor. Now this holorepressor molecule, they bind at the operator site. And as the operator site is blocked, so the RNA polymerase cannot transcribe the structural gene. Here heme act as a co-repressor. When there is excess amount of heme in the body, so the body tells the signal that there must be decreased synthesis of heme now. And how now heme regulates its own synthesis? Now this heme molecule act as a co-repressor. They bind with the aporepressor molecule. They form the holorepressor. This holorepressor bind at the operator site. Now the operator site is blocked totally. Now the RNA polymerase cannot move downstream and they cannot transcribe the structural gene of allosynthesis. So allosynthesis is decreased, so heme synthesis is decreased. Okay. Now students, we have covered the major portion of enzyme regulation. Next is your regulation by proteolytic trimming. Here some part, here some enzyme, the basic example is like some enzymes which are synthesized as a proenzyme forms, like the digestive enzymes which are synthesized as a zymogen or the proenzyme form. And some peptide bonds must be cleaved to activate it. So the basic examples are like the pancreatic enzymes which are synthesized as zymogen form. And now after reaching at a particular pH in the intestine, some peptide bonds are cleaved and now the proenzyme becomes activated. Like the example like is the chymotrypsinogen. So after reaching the intestinal juice, it becomes trypsinogen and it becomes activated. Pro-elastase to elastase. So this is another way of regulation where some portion, some peptide bonds are cleaved and the protein molecules like the enzymes are activated. This is another type of regulation. Now, the last portion, that is the regulation by the compartmentalization. So if you can recapitulate, you can see that there are many metabolic pathways which occur in cytosol. Some are occurring in the mitochondria and some are occurring both in the cytosol and mitochondria. So students, can you say some metabolic pathways which are occurring in the cytosol exclusively? Yes, they are like glycolysis, HMP shunt, fatty acid biosynthesis, etc. And some metabolic pathways which are occurring exclusively in mitochondria. These are asked in Viva. Which metabolic pathways are occurring in the mitochondria? These are like TCA cycle. Yes, electron transport chain. And another is beta oxidation of fatty acid. And can you say the metabolic pathways which are occurring both cytosol and mitochondria, like your urea cycle and neoglycogenesis? Some part of this metabolic pathway is occurring in the cytosol and some are occurring in the mitochondria. So what is the need of compartmentalization? So what is the need of compartmentalization? We'll discuss in the next slide. So yes, with this compartmentalization, the anabolic and catabolic pathways are well regulated. Okay, and the opposite pathways are spatially differentiated into different cellular compartments, like fatty acid biosynthesis occurring at the cytosol and beta oxidation of fatty acid at mitochondria, so that the regulation can be done well. If both are occurring at same place, then it is a haphazard regulation, but they are compartmentalized. So they are spatially differentiated. So the regulation will be much easier. And this compartmentalization necessitates the different transporter across the membrane. What is the meaning of this line? One example I am giving you, like the fatty acid beta oxidation. I have told that fatty acid beta oxidation occurs in the mitochondria and the biosynthesis occurring in the cytosol. So the long chain fatty acid, the free fatty acid which are in the cytosol, and when they have two beta oxidized, so they must reach the mitochondria. How they reach? Via the transporter, carnitine transporter, acyl carnitine transporter. So here, this compartmentalization, this necessitates the different transporters across the membrane. So with this we have covered the all type of regulations. The regulation of enzyme activity can come as a short note or can come as a long question. So you should read or you should read it well properly, and you should write it in 5 subtopics, like is your allosteric modification, then is your modification by covalent modification, then is your regulation at the genetic level, then is your compartmentalization, and third is your proteolytic, and the last one is your proteolytic trim. Okay,